Okay, so welcome back to again the active solid transport. So till now we discussed about the P class pump, right? So in this lecture we'll talk about the F and V class. So as you remember, like we have talked about the alpha and beta structure of the P class pumps, right? Wherein which we have discussed like alpha 2, beta 2, and हमने वहाँ पे discuss किया, correct? Now in this case, let's talk about the F and the V class pump. And later we'll talk about the ABC superfamily. Now, where ABC stands for ATP binding cassette. Okay, this is really very interesting, especially with respect to your multi-drug resistant bacteria and the cancer cells. We'll talk about this in detail. Just see the structure of all. बहुत ही different है. Now, have you seen this structure anywhere during your MSc, BSc?
Mitochondria.
Okay, so where have you seen this structure?
Mitochondrial membrane.
Correct, mitochondria. So there we have talked about the ATPases, that is which generates ATP, correct? So that is the same thing which we are going to discuss now. Okay, so let's talk about the F-type ATPases. These pumps, they possess membrane domain, transmembrane domain F0 and a peripheral domain F1. Okay, अब यहाँ पे क्या है? This is actually F0. Sorry, not F0. It is FO. But अंग्रेजों में ऐसे क्या कहते हैं, ऐसे O0 का कंफ्यूजन ना हो करके इन्होंने ऐसे दिया है, ठीक है? O because if the antibiotic by name oligomycin is there, so this can inhibit this particular transmembrane domain, इसलिए उसको O दिया है, but F0 पढ़ेंगे, ठीक है? Since उसको 0 पढ़ रहे थे, तो we have given this domain as F1. Okay. So they transport only protons down the electrochemical gradient for the synthesis of ATP from ADP and PI instead of hydrolysis. So it is a ATP synthesis. Okay. Now, जो mitochondria में हम हमेशा से पढ़ते आ रहे हैं, like the electron transport chain, and in that we have discussed about complex 1, 2, 3, 4, and 5. So this is the fifth complex, the ATP synthase. Okay. Now, okay, this I just mentioned now that O indicates the oligomycin. Okay. And it's a peripheral protein. One indicates that it was the first of the several factors from the mitochondria. Anyways, not required. Okay. So this is your structure. Now ये membrane में है यहाँ पे. Now माइटोकॉन्ड्रिया का आउटर मेम्ब्रेन हो गया, माइटोकॉन्ड्रिया का ये क्रिस्टे वाला हो गया, राइट? एंड हियर वी हैव गॉट द इंटरमेम्ब्रेन स्पेस. दिस इज cytoplasm. This is intermembrane space. This is matrix. Okay. Now, how the pump is hanging? ऐसा, something like this. Clear? Now, F class proton pumps. So in, of course, when we talk about mitochondrial P class pump, this is something which is similar to the bacterial proton pumps also. Okay. So bacterial plasma membrane, inner mitochondrial membrane, and thylakoid membrane of the chloroplast. So they all, wherever it is present, it is your F class proton pumps. Now thylakoid membrane of chloroplast, wherein which when we will talk about the photophosphorylation, जहाँ पे आपका फोटोसिंथेसिस में जो एटीपी बनता है, दैट इज विद द हेल्प ऑफ द लाइट, राइट? दैट्स व्हाई इट इज फोटोफॉस्फोराइलेशन. वहां पे भी एटीपी सिंथेसिस हो रहा है. सो दैट इज अगेन एफ क्लास प्रोटोपम्प्स इन द थाइलाकोइड मेम्ब्रेन. ना इनर माइटोकॉन्ड्रियल मेम्ब्रेन में क्या होता है आपका? ऑक्सीडेटिव फॉस्फोराइलेशन, दैट इज एटीपी सिंथेसिस. एंड बैक्टीरियल मेम्ब्रेन में तो हम उसको नॉर्मली हम H+ या P+ प्रोटॉन पंप्स कहते हैं, ठीक है? ओके. सो नाउ व्हाट इज हैपनिंग हियर इज you have got the membrane. Okay, this is a matrix intermembrane space. So what we have seen is that when the proton gradient is formed, यानी कि यहाँ पे H+ है, if you remember, with the help of the complex 1, 2, 3, and 4, eventually what is happening? The H+ are transported to the intermembrane space. Now you have got the high concentration of intermembrane space here. Sorry, you have got the high concentration of H+ in the intermembrane space. So what had happened here? यहाँ पे low है, यहाँ पे high है, तो proton gradient आपका बन गया है, correct? Now, So when H plus is moving from intermembrane space to your matrix, this gradient, okay, is converted into the mechanical energy. Okay, this mechanical energy results in the rotation of this gamma subunit, which is connecting between the F0 and F1. Correct? Now what is happening? This conversion is this rotation is generating energy. That energy is used to make ATP from ADP and PI. Clear?
Yes, ma'am.
Okay. So later when we talk about the mitochondria and this, you can just combine both the thoughts, like both the topics, and finally you can understand how exactly the ATP synthesis is working. Okay. Now this is the top, what to say, the top view of your pump. So what do we see here? You have got alpha ADP, beta ADP, alpha ATP, beta ATP, alpha MT, beta MT. अब आप सोच रहे हो कि ये क्या है? तो अभी हमने क्या देखा? H plus यहाँ से आपका यहाँ जा रहा है, ठीक है? And these are the regions where what is happening is that ADP is coming and binding. ठीक है? That energy on rotation is taken up by the ADP and phosphate to make ATP. अभी कैसे हो रहा है? See, alpha पहले आपका alpha beta empty है, okay? So this your ADP comes and binds. Getting the point? अब आप ये structure देखो यहाँ पे alpha beta है, okay? So now it is empty. Now it is bound to the ADP. So when that rotation is happening, this rotation I was talking about, right? Gamma. So gamma में जब ये rotate होगा, so when it is rotating, okay, so the energy release will be used to make the ADP into ATP. Correct? So now there is a stage where it is also having ATP, it is having ADP, and now it is empty. Next rotation में क्या होगा? ये ATP release होके बाहर चला जाएगा. By the time here you will have ADP and here ATP, and this will be empty. Next rotation, what will happen? In the next rotation, again another rotation had happened, right? So this ATP वाला ये अभी आपका empty हो जाएगा, यहाँ पे आपका ADP आ जाएगा, and this will be your ATP. So each rotation, one ATP is released. Clear? थोड़ा सा idea? Are you able to imagine, guys?
Yes, ma'am.
थोड़ा सा और समझाते हैं, ठीक है? So already said that H plus is resulting in the gamma to rotate within the AB alpha beta head. Okay, this is the same figure. Now let's talk about the three conformation. So first क्या हो रहा है आपका? Can you see ये आपका empty है, यहाँ पे आपका ADP है, यहाँ पे आपका ATP है. Okay, take any scenario, take any scenario. So let's talk about this. So यहाँ पे क्या हुआ? आपका H plus आया, ठीक है? There was a rotation. That rotation resulted in the release of this ATP and binding of ADP here and formation of new ATP here. Again, what you see? इस पे ध्यान दो, जो आपका रेड में हाईलाइट है, ठीक है? उसमें ध्यान देना. So empty, ADP, ATP. Now rotation had happened. So what had happened to that stage? जो ADP था वो ATP हो गया, जो ATP था वो आपका खाली हो गया, जो खाली था वहां पे आपका ADP आ गया. Next rotation what had happened? This जो सबसे पहले आपका यहाँ पे ADP था, अभी वो ATP बन के रिलीज हो गया है. जो ATP था वो अब आपका ADP है, एंड यहाँ पे जो था यहाँ पे आपका ATP आ गया है. Okay, so this is just the cycle. आप कहीं से भी शुरू करो, eventually what is happening? It is binding the ADP and phosphate, stabilizing the transition state for the phosphorylation of the bond. Okay, the formation of the phospho anhydride bond जो बनता है ADP and P के बीच में, right? And eventually resulting into the ATP. अब हर काम के लिए हमें ATP लगता है, मगर ATP बनाने के लिए हमें प्रोटॉन ग्रेडियंट लगता है. Clear? I think this figure is more clear than the rest. Yes. Is it clear to you, Akshay, Tanvi?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. So I'm not asking you to repeat, otherwise late ho jayega. But I believe that it is clear to you all, and it is important also to understand. Okay. This is just the same figure. Okay. दूसरा मुझे ऐसे मिल गया, so I thought of just putting it so that it is clear to you. So again you can see open means यहाँ पे empty है. So ADP will come here. Proton flux, again rotation is happening. Okay, 120 degree. Since it is complete 360 है ना, 360 को हमें 3 बार में divide करना है. So how much we are getting 120? So that's why it is 120 rotation by the gamma subunit. तो ये थोड़े ऐसे छोटे छोटे पॉइंट्स हैं जो आपको एग्जाम में पूछ सकते हैं. सो व्हाट इज हैपनिंग नाउ, जो ओपन था अभी यहाँ पे एडीपी आके बैठ गया, पीआई आके बैठ गया, रोटेशन हुआ, दैट रोटेशन was used by the that energy was used in order to make this phosphoanhydride bond between the ADP and the phosphate. ठीक है, and eventually it will result in the formation of the ATP. Okay. Now the third class we can talk about the V class pump. So where do you find this V? You can find on the vacuole. और vacuole कहाँ रहता है आपका? फूड वैक्यूल, गैस वैक्यूल और प्लांट्स में प्रेजेंट रहते हैं, करेक्ट? सो ऑल दोज एरियाज द V class pumps are present. Okay, so they are similar, and they are similar in structure and function of the proton pumping to the F class pumps. Okay, but the proton transport is against the proton electrochemical gradient. Now again here V0 V1 is there. It has got 5 and 8 subunits. ये कोई नहीं पूछेगा. Found in the vacuole membranes and the lysosomes, endosomes हो गया, macrophages हो गया, osteoclasts and the renal intercalated cells. Okay, looking at the diagram, थोड़ा बहुत तो it looks like a F class pump, correct? But it has got more subunit as compared to the your F class pumps. Okay. वहां पे क्या था आपका, if you remember, it was H plus from the intermembrane space to the matrix side, correct? Here it is ulta. Here it is from the cytoplasm to the lumen, and eventually again it results in the rotation of this shaft. Okay, just that it is opposite of the proton gradient. Now let's talk about the function. ये थोड़ा सा important है, and I have added these points because it was asked in the exams. So the plasma membrane V ATPase is involved in process such as pH homeostasis and the tumor metastasis. Okay, अगर अभी यहाँ पे मैं एक example देती हूँ आपको. When you how to say, when you feel hungry, you can feel a kind of burning sensation, right? आपको पता चलता है कि आपको भूख लग रही है. Agree? So that hungry means what? The pH is getting increase or decrease. 1 second, guys. Yeah. So sorry. So what I was explaining was that when you feel hungry, there is a burning sensation, and that time what is happening? If you know, in the stomach, okay, in the stomach lining, your pH is increasing or decreasing. क्या हो रहा है वहां पे? In the stomach, whenever you feel hungry,
Decreases.
correct, your pH is decreasing, correct? So ये चीज जो है वो कौन लाता है आपका? H plus ATPases. Okay, so they are responsible for making more concentration. तो जितना ज्यादा आपका H plus आएगा उतना ज्यादा आपका pH decrease होगा, correct? And that's why these H plus ATPs are coming under this category. अब वो कैसे काम करते हैं, this is what we will discuss in this topic. Okay, yeah. So V ATPases in the acrosomal membrane. This is another example. One example I have already given. This is another one. So in the acrosomal membrane of the sperm, acidify the acrosomes. This acidification activates proteases, which is required to drill through the plasma membrane of the egg. So guys, I hope you know what is acrosomal membrane of sperm. अगर आपने देखा हो, so we have got the egg over here. Okay, so it has got ऐसे what to say, a kind of protection so that it doesn't get because already it is in the harsh condition in the uterus, right? So what is happening over there? The sperm is coming, I say something like this. Okay, so this portion has got this V80Ps class. So this is going to help in, making the pore into the wall of the egg so that the DNA can be pushed inside. Okay. V80Ps in the osteoclast plasma membrane. What is osteoclast? It is your bone cells. Okay. Now, it is also present in the osteoclast plasma membrane in order to pump protons onto the bone surface, which is helping in the bone reabsorption. यानी कि क्या? यहाँ पे हम calcium 2+ की बात कर रहे हैं. ठीक है? Now, in the intercalated cells of the kidney, V80Ps pump protons into the urine, allowing bicarbonate reabsorption into the blood. So here we are talking about the reabsorption. We need to make the to maintain the pH of the blood. Now, in addition, there are other variety of biological processes such as toxin delivery, viral entry, apoptosis, regulation, etc. Everything are coming under as the important roles of the V class ATPases. ठीक है? तो अभी हमें आइडिया मिल गया like how much it is important for us. Now let's talk about the lysosomal V type pumps. Okay, so when endosomes bud off from the plasma membrane, the V class pumps increase the acidity of the endosomes. Now why? Because एंडोसोम्स में ही इवेंचुअली जो भी आपने खाया है या जो भी ऐसे समथिंग इज engulfed by the cell or whatever it is, finally it goes and gets fused with the lysosome, right? And the degradation starts. Okay, so they all have got this V class pump. Clear? Now let's take the example of cancer cell over here. So these are your V type pumps present here, H+ वाले present है. ठीक है? Now what do we see here? आपको फिगर से क्या समझ रहा है? Anyone can guess? Anyone who can guess क्या हो रहा है यहाँ पे? Yes. नहीं? Anyone?
Is it releasing the degradative enzymes?
Yes,
outside?
correct. But then add more to that. So how the V class pumps are helping? See, if you see this structure, ये जो blue वाला है, they are the forms of the V class pump. यहाँ पे H+ pump की हम बात कर रहे हैं, ठीक है? So when you increase the pH inside of the lysosome, what is happening? Not increase, sorry. When you increase the H+ concentration, the pH is decreasing, correct? And that decrease in the pH is activating your proteases. So from immature, the green ones are the matured ones. Okay. So this is getting activated. Now, once these are activated, or here they are here they are exocytosed out. Okay, यहाँ पे उन्होंने transfer कर दिया. अब proteases क्या कर रहे हैं? They start targeting their tissues. अब वो क्या करेंगे? सारा chop off करके whatever the nutrients available to these target tissue, they will take, and cancer cell will get all the nutrients required for its survival. Plus, it is blocking the apoptosis. Hence, the cancer cell just eats and grow. So another takeaway message is that if you could block the V80Ps in the cancer cell, you could target the cancer cell death. अब समझ रहे हैं थोड़ा सा? Yes. So there are many ways by which you can actually target cancer cell. The problem is that these lysosomes, these H+, or this particular type of V80Ps are also present in the normal cell. So the main question lies is in the recognition of the medicine or the drug which is going to be very specific to the cancer cell and not affecting the normal cell. But if you know the stats, we have seen that along with the cancer cell, the normal cells are always affected, and hence the cancer patients, people, they start losing kidney, liver function, they start losing hair, etc. Okay. Now coming to the human disease, if suppose the V class pumps are not functioning properly, you have got the osteoporosis, then the bone fractures, because of course there is no reabsorption of the bone. So what will happen? The bones are going to become brittle. Then the other one is the renal tubular acidosis. So pH, urine pH goes below 5. So the people who have got the low pH, then for them urinating is also very difficult. Then we have also X-linked myopathy with excessive autophagy. Okay, so these are the different diseases which is associated with the mutations in the V class pumps. Okay, yeah. That's all, guys. So this is what we have covered today. The P class, F class, and the V class ATP, active transporters. Okay. So I'll just stop here and then we can discuss if any questions are there.